Hello, everybody. My name is George, and this is going to be a quick walkthrough through today's lab, um, in which you will be extracting genomic DNA from a hair sample, and you'll be pretending to be a forensic scientist who discovered some hair at the crime scene. And this lab was developed by Dylan during his work. Last summer in our lab at Stanford. And this whole effort is supported by the National Science Foundation. So, right, the first step will be to prepare your hair samples, and you can pluck hairs from your arms or your eyebrows or your scalp. We'll need about 5 hairs, 5 good hairs, and what's important is that your hairs have nice follicles, and the follicle is the part at the end which contains lots of cells, lots of skin cells. And you probably want to do this on a clean piece of paper so you see what you're doing, and you want to work quickly so the hair follicles don't dry out fully. So once you have your hairs, the next step will be to trim them, right? So what we care about is the follicle, which is at the tip here. The rest of the hair shaft is not really important. So you'll trim it to about 3-4 millimeters, sort of a small piece. You can use a razor blade or scissors, like in this case, and then you keep the bit that has the follicle and you put it in a Eppendorf tube, one of those little tubes, and you'll collect 5-6 follicles like that. And it's important that you label your tubes. That way you won't confuse them with your neighbors. Right. So after you collected the hairs, the next step is to push the hairs all the way to the bottom of the tube. And this is important because we'll add liquid, and we want to make sure that the hairs are immersed in liquid. You can do this with the tip, with a pipette tip. And hopefully you can get it all to the bottom. Right, there you are. Another way, right, so once you have the hairs at the bottom of the tube, the next thing that we're going to do is to add this 10% Chelex slurry. So what the slurry is, these are tiny beads that bind or capture metal ions like magnesium. And the reason this is important is because magnesium is required for the activity of nucleases, and these are enzymes in our cells that cut DNA. So when you capture the magnesium, the nucleases cannot function, so they don't cut the DNA. So in a way, Chelex helps preserve the DNA by inactivating these nuclease enzymes. So you will have a Chelex resin in a tube. Dylan here has a very large tube, but your tube will be smaller. So the first thing you want to do is you want to resuspend this slurry by inverting the tube, or you can pipette up and down until you get the homogeneous mixture. And then you pipette 50 microliters of that into the tube that has your hair samples. And this is what Dylan is doing here. Right, so get it in the tube with your hair samples. And it's important that the hair samples are fully submerged in the liquid. And you can use the tip of the pipette to make sure that that's the case. Right, so here they are. They should look something like this. You can see the Chelex beads are sort of this whitish stuff at the bottom. And then you just have water on top. So the idea is we will add an enzyme to this mixture. The enzyme is called pertinase K, right? And pertinase K will digest the cells of the hair follicles and will liberate the DNA into the solution. If you have trouble getting the hairs to the bottom of the tube, something else you can do is you can take these tubes and you can centrifuge them. All right, so what you have here is you have these plastic adapters in which you can put your tube with your hairs, with the Chelex, and spin it down, and you get it all to the bottom. And this is optional if you have trouble with the pipette tips. And in order to spin these tubes in the centrifuge, you might have to make your own makeshift adapters. So for that, you take one of these larger tubes. These are 1.7 ml tubes. You just cut off the caps. And then you can place your smaller tubes that have your samples into these caps, into the centrifuge, right, like that. And it's important to make sure that your centrifuge is balanced. So you have one tube on one side, and on the opposite side, you have an identical tube. You can put some water in there, or maybe even an empty tube. That way, when the centrifuge spins, it doesn't go out of balance. And this is a quick 30-second spin at is it 5,000, 6,000 RPM? Yeah, 5,000 RPM. Okay. So after the spin, you are ready to add Pertnase K to your sample, right? So the idea is that Pertnase K is an enzyme that chews up proteins, and your cells are made of proteins. So when Pertnase K chews up the cells, it releases the DNA into solution, which is what we need. And to do this, you will get a Pertnase K aliquot, a little tube that has enough enzyme, and the enzyme is normally stored at -20 in the freezer. And you'll get that from your teacher. You will set your pipetter to 2 microliters and pipette out 2 microliters. The tube should have about 4 or 5 microliters, so there's a little bit of extra. And you carefully add that to your tube with Cheelex slurry and with the hair. Like that. Make sure you add all the liquid. That's it. Okay, so then you've added the Pertnase K to your sample. The next thing is we need to mix. And to get good mixing, we normally use a vortexer. You can vortex for about 10 seconds, and this mixes the Pertnase K with your beads, with your Cheelex beads, with your hair, and it also mechanically begins to break up the cells in the hair follicles. And after you do this, you'll spin down the tube again just to make sure that everything goes to the bottom. Yeah, you need to make sure to put the cap. There you go. And again, 30 seconds, 5,000 RPM is sufficient for this. Right, so after you've done the mixing, now you're ready to incubate it. And we often do this very, very gentle mixing called plinking, where you hold the tube in one hand and you kind of plink the bottom of the tube, and this allows the Cheelex resin to be resuspended. Then you can put it in one of those rotating incubators, and you incubate for about 45 minutes. So during this time, the Pertnase K will slowly digest the cells and will get more and more DNA into the solution. If possible, try to place this incubator, this rotator in an incubator that's warmed up to 37 degrees, which is body temperature. And the idea is that Pertnase K works better at 37 degrees than at room temperature. Okay, and after this incubation, now you're sort of getting to the end of the lab. What we're trying to do now is we're trying to heat and activate the Pertnase K. So the Pertnase K hopefully did its job, but you don't want it to remain active, because if it remains active, then it will degrade the enzyme, the polymerase enzyme used in the next lab, right? So you want to use this DNA to do PCR, polymerase chain reaction, during your next lab, but if Pertnase K is around, it'll chew up the polymerase. So for that, if you incubate Pertnase K at 95 degrees, which is basically almost boiling, for 10 minutes, it will be denatured by heat and it'll be inactive. So that's what this incubation is about. And the one detail here is you want to add water to the heat block to make sure that there's efficient heat transfer from the heat block to your sample, which is in the plastic tube. So this will take 10 minutes. And then you are ready to isolate the extracted DNA. So during those 10 minutes, what you can do is you can prepare this column. It's almost like a little filter, which will capture the Chelex beads and will keep them separate from the DNA solution. And the idea is that we don't want any Chelex beads remaining in the DNA sample because it will continue chelating, capturing magnesium ions, and magnesium ions are important for the function, for the activity of the PCR enzyme during the next lab. So we want just clean, pure DNA solution without any Chelex. So what Dylan is doing here is he cut the tip of the column, just to back up a little bit. Right, the column has a cap, this is a sealed end. You can use a sharp razor to cut that end, and now the column can be used to basically filter out your Chelex beads. There you go. And then you can place this column. The column is dry and empty. You can place it in a pre-labeled Eppendorf tube, and these are 1.7 ml Eppendorf tubes, so the slightly larger ones. And you can use the red cap to cover it up when you have the sample. Okay, so now this tube is ready for collection. So after the 10-minute incubation at 95 degrees, the goal is to now transfer all the sample into the column in order to isolate the Chelex beads from the DNA. So you take all of it and you pipette it inside the column. And you can do it in a couple of steps just to make sure you get all the sample. There you go. So now you can take this, cap it to make sure that you don't lose anything. And this is what it looks like on the inside. You have a slurry of Chelex, maybe some hair, and liquid, which contains your DNA. So in order to separate the DNA from the Chelex, we take this tube and we centrifuge it. Again, for 30 seconds at 5,000 Gs. The only thing that you have to be careful about is now you need to balance the centrifuge, and the balancing is a little bit different because the tube is bigger. And so you can't spin it with only one tube, you need to make a balance. So in the one thing you can do is you can take the tube that has your sample, weigh it, see how much it weighs, and then you can make another tube and put enough water to reach the same mass, and you can use that as a counterbalance. And then you load it in the opposite portions of the rotor, close the rotor, and spin it. So this will push the DNA containing liquid through that column, which has a filter at the bottom, and all the liquid will pool in the collection tube, and all the Chelex resin will stay in the column. So once you're done centrifugating, you take out your tube, and this is what you see. You see clean buffer, clean liquid containing a DNA, and then you see the dry Chelex resin and the column. We don't need the Chelex anymore. You can throw it away and just keep the tube, cap it, make sure you label it with your name, your team name, and this sample can be stored in the freezer at -20 degrees for a couple of weeks until you get to the PCR lab. So this is the end of the forensic DNA extraction laboratory, and hopefully you prepared a good sample, and we'll see how good the sample is in the next lab. Good job.